Hi, my name is Heather Ald, and this is my talk on audience effects on male mate choice in the Trinidadian guppy. This presentation will supplement the poster that I gave at the Animal Behavior Society meeting in Boulder, Colorado, or if I can get a chance to talk to you and you're interested in the work that I'm doing, this video will also explain some of the work. So to start, sociality. Most animals are social at some point of their lives, whether it's during foraging or during mating, or simply living in a group. And this ranges from all sorts of taxa, from insects to birds, fish, and mammals. When living in a social environment, individuals can acquire non-genetically acquired information. So there are a couple types of non-genetically acquired information that you can receive. One being private acquisition, so that's if you search out things and learn them on your own. Another way to gather information is through social information. There's two kinds of social information. One is signals or direct communication. So that's when one individual is directly trying to communicate with another and trying to convey some sort of information. The other option is cues or inadvertent social information. But this is information that is gathered by individuals who are not necessary, who the sender is not trying to send them information. So they're watching someone and getting information that way. So there's two different kinds of inadvertent social information, one being location cues, so by seeing other individuals, seeing where they are, and the other is public information. And public information is information that is indicative of the quality of the other individual, or something, how well they're doing. So it could be their mate choice, it could be where they're foraging, or where they're seeking shelter. So there's several social phenomena that can happen with this acquisition of non-genetically acquired information, some of which was touched on the previous slide, but I'm going to outline it a bit more clearly on this slide. So the first is eavesdropping, and that's how individuals acquire this information from public information. So in this photo here, there's an interaction going on between these two birds. Now I'm not sure what's happening. They could be fighting, they could be playing, but whatever is happening, individuals around them, such as this guy here, or girl, can extract information from that scenario. So that bird may be able to say, oh, this bird is strong, is fighting another bird, or that bird's aggressive, I do not want to go near that bird, and such forth. So one thing that can happen with social eavesdropping is copying. So one of the options is an individual can copy the choice of another. So if they see an individual foraging a certain food patch, they can go to that food patch and forage as well if that individual looks like they are doing well. Another option is mate choice copying. So individuals can copy the choice of another mate. And this can lead to an audience effect. If an individual knows that it's being watched and it's in a situation where being copied or giving information may not be to its benefit, such as in mate choice copying, where that individual wouldn't necessarily want a bunch of other males copying his mate decision, as then it could lower his potential reproduction with that female. We have something called the audience effect. So the audience effect is where an individual or individuals behave differently when there's an audience around. So in this situation, there's one polar bear here, and him sitting there watching, or her sitting there watching, could be affecting the way these two bears are behaving. So traditionally, people thought of mate choice as individual between two individuals. But there's been more research showing that mate choice can be non-independent. Most of this work has been done in females, but recently there has been work done on non-independent male mate choice in several species of fish, including the mollies and the pipefish as well. And recently, some of my work, we've showed mate choice copying in the male guppy. This is still in preparation. So the model species that I'm working with now is the Trinidadian guppy, and this is a really great species for this kind of work because they form dynamic mixed-sex shoals. Both males and females are promiscuous, and they have a non-resource-based mating system, and both males and females exhibit mutual mate choice. The fish that I used were collected from the Upper Aripo Naranjo tributary in Trinidad, which is in the northern mountain range. And the question that I asked for my first experiment is, does an audience of potential sexual rivals affect a male's mate preference? And I predict that males will show a reduction in preference for their initially preferred female in the presence of a potential sexual competitor. So the way that I measured their preference was using a dichotomous choice test. So what happened was there was two females on either end that differed in length by about 20 to 25%, and males are shown to prefer larger females overall. So the male was placed in a central cylinder inside the tank and allowed to observe the females for 10 minutes prior to being removed. And once he was removed from the cylinder, he was able to swim into the preference zones of the other females, which are denoted by the dotted lines. And the time he spent in each preference zone was recorded as a proxy measure of his preference for that female. After 10 minutes, gates were put in and blocking the females, and the male was replaced into the cylinder. At this point in time, the sides of the females were switched to control for any potential side bias of the male. Once the female sides were switched, the opaque partitions were removed, and again the male was given 10 minutes in the tank. So more specifically, I measured first his initial mate preference. So it had a small female and a large female in the dichotomous choice test with the initial mate preference. And then in the second preference test, there was another audience male or heterospecific fish in a cylinder during the test. So there was 4 treatments. 3 were done previous altogether, and then the 4th treatment was done afterwards as a control. So treatment 1, the control, had an empty cylinder. Treatment 2 had 1 conspecific audience male, and treatment 3 had 2 conspecific audience males. Afterwards, treatment 4 had 1 heterospecific fish, the zebrafish. And this 4th treatment was to control for the disturbance effect. So what the results show is when there was a potential sexual competitors present, males showed a reduction in their initial mate preference. So there is a significant difference between both the first control and the heterospecific control and the 2 treatments where there was a conspecific sexual rival. There was no difference whether there was 1 male or 2 males. Similarly, when there were potential sexual competitors present, males showed an increased likelihood of reversing their mate preference. So this leads me to the second question. Does the relative quality or attractiveness of the audience male affect the magnitude of the audience effect? And so I predict that the audience effect will be greater when the audience male is more attractive. And the 2 measures that I used for attractiveness were length and body coloration of the black and orange spots. So in all there was, I combined these characteristics for smaller body size, similar, larger, coloration, less, similar, more, and a combination of those. So small and less colorful, similar, similar, large, more, smaller, more colorful, smaller, similar coloration, etc. So this was the result of the preference difference score with the color score difference. Now, as you can see, there really isn't much of a pattern here. R squared is very, very small, and it is not significant. Using the body length difference between the focal male and the audience male, the R-squared value is still really small. However, P is 0.02. It is significant. Now, after I printed my poster, I had an idea about how to divvy up the categories. So I divvied up the categories, finding the mean of the differences in body length and the mean of the differences in color difference. And then using a standard deviation on either side of the middle, I found the extremes on the large end and the extremes on the small end, the extremes on the more colorful and extremes on the less colorful. Now, looking at this and having it divided into 9 treatments, which are listed on the side, there does appear to be a relationship. So preference difference score, I want to point out the most different are when the audience male is small and dull. The focal male actually increases his preference for his originally preferred female by about 6%. And when there's a large, bright audience male, he decreases his preference by about almost 30%. So that's about a 36% difference. And I find this really quite interesting. But it's still preliminary, and I have more work to do on this to really sort that out and to display it properly. But it's really interesting to look at. So the implications of this. So we do know that there is an audience effect, and this can have implications on sexual selection models as well as social networks, in particular male-male relationships. So if there is this changing of mate preferences whilst other males are around, is this going to affect how males associate with each other? Are they going to associate with males who are more attractive than themselves or less attractive than themselves? So there are questions in future work. So some of the questions that I still have are, what are these other sources of variation? When we look at those graphs, there is not a lot of variation explained by the male phenotypes alone. So perhaps another source of the variation is the behavior of the audience male, which is something that I didn't record. It could also be influenced by the behavior of the females, or the personality of the females, or the personality of the audience males. It would also be interesting to look at interpopulation comparisons. So I would like to acknowledge Elisa Miller for help with data collection, Dr. Grant Brown and Chris Elvidge for fish collection, Professor Indar Ramareen for logistical support in Trinidad, the Director of Fisheries in the Ministry of Agriculture, Land and Marine Resources of Trinidad and Tobago for permission to collect wild guppies. And this work was funded by an NSERC research grant to Professor Jean-Guy Godet. And if anyone would like to further talk about any of this work, I would be thrilled to do so, and you can contact me at heatherald@cmail.carleton.ca. Thank you so much.